Hello everyone. So the next topic which we will be discussing here is about the epigenetics. Now, what is the definition of epigenetics? So epigenetics means above or on the top of the genetics. It refers to the external modifications to DNA that turn genes on or off. So these modifications, they do not change the DNA sequence, but instead they will affect how the cell will read that sequence. So the term epigenetics refers to the heritable changes in gene expression, active versus inactive genes. So that does not involve the changes to the underlying DNA sequence. A change in the phenotype without a change in the genotype is there. Now comes the introduction. So epigenetic changes helps to determine whether the genes are turned on or off. They influence the production of proteins in the cells. And this regulation helps ensure that each cell produces only proteins that are necessary for its function. So if I take an example here, the proteins that promote the bone growth are not produced in muscle cells. So the patterns of the epigenetic modification vary among individuals in different tissues within individual and even in different cells within a tissue. So epigenetic change is a regular and a natural occurrence but can also be influenced by several factors including age, the environment, lifestyle and the disease state. So epigenetic modifications can manifest as commonly as the manner in which the cells terminally differentiate to end up as skin, liver cells or brain cells, etc. So now comes the history of the epigenetics. So the term epigenetics, which was being coined by Cornard H. Waddington in 1942, was being derived from a Greek word. So the Greek word was epigenesis, which originally described the influence of genetic processes on the development. So these were the two scientists who started the study of the epigenetics. So here is a picture of both the scientists which is being. So the system of epigenetics has been divided into three parts, that is the DNA methylation, the histone modification and the non-coding RNA. So first we will discuss about the DNA methylation. So what is DNA methylation? So DNA methylation, it is a biological process by which the methyl groups, they are added to the DNA molecule, and methylation can change the activity of a DNA segment without changing the sequence. So when located in a gene promoter, DNA methylation typically acts to repress the gene transcription. So in case of mammals, DNA methylation is essential for the normal development and it is associated with a number of key processes that may include the aging, carcinogenesis, chromosome inactivation. So these are some of the this is some of the overview of the DNA methylation. The next is about the histone modification. So histone in biology, the histones, they are highly basic proteins which are abundant in lysine and arginine residues that are found in the eukaryotic cell nuclei. So they act as spools around which DNA binds to create structural units called the nucleosomes. So nucleosomes in turn are wrapped in 30 nanometer fibers that form tightly packed chromatin. So histones, they prevent DNA from becoming tangled and they protect it from the DNA damage. So in addition to this, histone plays an important role in gene regulation and DNA replication. So without the histone, unbound DNA in chromosomes would be very long. For example, each human cell has about 1.8 meters of DNA. So if it completely stretched out, however, when wound about the histones, this length is reduced to about 90 micrometers or 30 nanometer diameter chromatin fibers. So this was about the histone modification. The next is about the non-coding RNA. So the non-coding RNA, it is an RNA molecule that is being not translated into a protein. So the DNA sequence from which a functional non-coding RNA is transcribed is often called a RNA gene. And abundant and functionally important types of non-coding RNAs, they include the transfer RNA, ribosomal RNA, as well as small RNAs such as microRNAs, siRNAs, piRNAs. So these are some of the non-coding RNAs. So this was about the non-coding RNA. The next comes is the mechanism of the epigenetics. That how is the mechanism going on? So there are several types of epigenetic inheritance systems that play a major role in what has become known as cell memory. Note, however, that not all of these are universally accepted to be an example of epigenetics. So here what is happening is, this is an epigenetic mechanism in which there is a development, environmental conditions, drugs, pharmaceuticals, aging, and diet. So this is a chromosome. So from here, the one part of the chromosome is called as the chromatin. So chromatin, it has a DNA in it. So there is a process of DNA methylation process which we have already studied. So the methyl group, which is an epigenetic factor, it is found in some dietary sources and that can tag DNA and activate or repress the genes. So this is a methyl group. It has been added to this. So histones, they are the proteins around which the DNA can bind for compaction and the gene regulation. So this is a gene and this is a histone and this is a histone tail which is there, and the DNA is inaccessible, gene inactive. So the DNA is here is inaccessible. The next part is the histone tail. So what is happening is there is a histone modification which takes place. So there is a binding of epigenetic factors to histone tails which alters the extent to which the DNA is wrapped around histone, and the availability of the genes in the DNA is to be activated. So the next is in the histone tail, this is an epigenetic factor which is there. So health endpoints, cancer, autoimmune diseases, mental disorders, and diabetes. So these are some of the health endpoints of the epigenetic mechanism. So this was all about the epigenetics. These are some of the references.